Hello, Bisc students. Today I'm going to be covering Chapter 3, Prokaryotic Cells. So we're going to focus mostly on prokaryotes through this entire semester. We'll talk a little bit about eukaryotic organisms, but we'll mostly focus on prokaryotes like bacteria and archaea, among others. So first things first is we have to know what a prokaryotic cell actually is, right? And hopefully you recall from earlier chapters, Chapter 1 and Chapter 2, we mentioned prokaryotic cells versus eukaryotic cells, how they're different. And the major difference is the nucleus. In prokaryotic cells, that if you break that apart, it actually means pre-nucleus. So DNA is actually not found in a nucleus in prokaryotic cells. Where eukaryotic cells, eukaryote means true nucleus. So the DNA can be found within the nucleus in a eukaryotic cell. So the first life originated about 3.8 billion years ago, and it was prokaryotic cells, single-celled organisms that didn't have a nucleus. Now, obviously, none of us were around 3.8 billion years ago. But we have some pretty good scientific evidence that single-celled organisms arose around that time. Some key facts about prokaryotic organisms: they're unicellular, they lack a nucleus, and they also lack membrane-bound organelles. Membrane-bound organelles include things like what you may be familiar with in a human cell, like the Golgi apparatus, the endoplasmic reticulum, lysosomes, for instance. So these things are more towards what eukaryotic organisms have versus prokaryotes. And this is a really good image of a prokaryotic organism. This is the capsule, the outermost layer. Remember, we mentioned a capsule is simply the glycocalyx, the sugary coating on the outside. If it's tightly wrapped, it's considered a capsule. If it's loosely wrapped, then it's considered a slime layer. But here it's tightly wrapped around the cell wall, which is the orange-reddish color here, reddish-orange. That's your cell wall. And then there is a cell membrane, this light yellow one right around the edge here, and then the cytoplasm, and you have here the chromosome. Bacteria have a singular chromosome. It's circular. It's like if you take a rubber band like this, okay, and that's what the bacterial chromosome looks like. However, to save space, it twists and folds and twists some more and folds some more and twists some more and folds some more until it takes up significantly less space. But if you were to carefully unwind it, it would actually be one giant circle. Some other structures, ribosomes, they're there for protein production. Inclusion bodies we'll mention later on. And when we talked about proteins, we talked about flagella and how they're made up of the flagellin protein. Not all cells have capsules or flagella as well. Depends upon the organism, whether or not they have those structures. There are two terms we'll talk a lot about, and that's arrangement and morphology. Arrangement means how they're grouped together. Morphology means shape. So if we're focusing on shape, morphology, we can have organisms that are what we call monomorphic, mono meaning one, morphic meaning shape, so one shape, or organisms that are what we call pleomorphic, meaning many shapes. So there are some bacteria that can change the way their structures look to alter their overall general structure. So they can change things within the cell to alter their outer structure and the way they look. Vibrio cholera is a really good example of this. It can be found in many different shapes and arrangements. And we'll talk about some of these later on when we start focusing in on specific diseases. Prokaryotic organisms are generally really small compared to eukaryotic organisms. This is just showing you they can range from 0.2 to 750 micrometers in diameter, which is really small, which means we need a microscope to visualize them. Light microscopes are powerful enough, but we actually need some help. We actually have to stain the bacteria first to visualize them because they're that small. So we stain them first, and then we can see them under a light microscope. You could also use an electron microscope, but that it's a little bit more complex and not totally necessary in some cases. So there's a key here that's pretty important when it comes to size. We can apply it to eukaryotic organisms as well as prokaryotic organisms, right? So surface area to volume ratio is very important. So surface area is the area that can interact with the outside environment or with other organisms. The volume is the size. So let's say we have a cell that's like this big. Front is surface area, there's top surface area, side surface area, and then the back surface area. That's a ton of surface area. But look how big this cell is. It's huge. So there have to be ways for us or even prokaryotic organisms to decrease the size but maintain all that surface area. And one of the ways to do that, which is really great, is to fold. Okay? So you can see here I've folded this cell. It still has all the front surface area, top, side, back, but now it takes up significantly less space. Okay? This is really common in our digestive tract. The cells fold in on themselves because it takes up less space but maintains a high surface area. This allows for things to be transmitted in and out of the cell pretty quickly, allowing transfer of carbohydrates or lipids, proteins, things along those that kind of way in that mechanism. So maintaining a high surface area to or low volume ratio is really important. Talking about shapes and arrangements, so here are some arrangements. You have single, diplo, dies 2. Strep or strepto means chain, or staph or staphylo means cluster. Now, when you say something is strepto, let's say streptococcus. Coccus is a round cell, a round cell. We call it coccus shape, so it's round. And if you have them in chains, typically we would have to say there's about 5 or more to be considered a chain. So strepto, and you can see 1, 2, 3, 4, 5. So if I were to say streptococcus, that tells me that there are 5 circular cells in a chain. If I were to say staphylococcus, I would say there is a cluster, what we call a grape-like cluster of spherical cells, of round cells. There's also a shape called bacillus, or rod-shaped. Basically, they're elongated. And again, you can have strep. In terms of streptobacilli, which is chains of rod-shaped bacteria. Palisades. This is where they're kind of just connected on one end to each other. And then there's some other really key shapes. Some that we'll see later on, and some that we really won't focus on. We won't focus on stella or filamentous, some pleomorphic, but not really. Spiral and spirochete we will. Vibrio we'll mention as well, and coccobacillus. So we'll get to a lot of these as we start getting to diseases of the organ systems, and we start focusing on specific bacteria. So, unlike humans, when we want to undergo replication and copy our cells to make new ones, let's say our skin cells, they're replaced about every 2 to 3 weeks. So, in order to replace those cells, we have to make new ones. We use mitosis to do that. Bacteria uses a process called binary fission. It is pretty similar to mitosis in that the end goal is to make 2 identical daughter cells. Genetically, they're the same, but it's a little bit more simplistic than mitosis. You can see the steps here. DNA is copied. That's because we want both cells to be identical genetically. Cell grows. One copy of the DNA goes to one side, one copy goes to the other side. Down the middle, it splits. There's a partition that forms. And then that partition, or what we call a septum, will eventually cleave the cells in half, and you end up with 2 new daughter cells that are genetically identical. And here's a good example of this. This is a figure in your book 3.7. Shows you prokaryotic chromosome. It copies itself, sends them to other opposite ends of the cell. Partition forms. Eventually the cell completes partition and it breaks them in half, and now you have 2 identical daughter cells, genetically identical daughter cells. Size. They try to keep them roughly the same size. They try to divide them pretty evenly, but it's not always. It doesn't happen all the time. Sometimes one cell is a little bit bigger than the other, but genetically, they should be identical. We've mentioned capsules. We've mentioned the cell wall. We also need to really focus in on the plasma membrane as well, or the cell membrane. So the cell membrane is made up of phospholipids. We actually talked about that in the previous chapter. They have a phospholipid bilayer to form the cell and everything that's inside the cell. And then outside that, we have the cell wall. Now, the plasma membrane is simply 2 molecules thick, and it can let things in or prevent things from entering the cell. Depends upon what you're trying to, or what the cell is trying to move in and out. Cell wall just lies outside that plasma membrane, and we'll talk more about that in a bit. This is what the phospholipid bilayer looks like. It's this thin, flexible layer. It actually is really flexible, so the cell can stretch and kind of bend at certain locations. And it is a very selective barrier, meaning certain things can bypass the cell wall and or cell membrane and go right through. Other things cannot. Some of the molecules that can go through a phospholipid bilayer just naturally are mostly what we call non-polar substances, things that don't have charges. So a good example are lipids, fats. They do not have charges, therefore they can bypass the cell membrane pretty easily. And then there are other molecules that need help. For instance, if you have an ion, which is basically an atom, whether it has an extra electron or an electron's been removed, right? So a good example of that would be sodium, NaCl. Chlorine rips an electron away from the sodium, so when salt dissociates, when it separates, you get Na plus and Cl minus, because Cl, chlorine, gained an electron, which electrons are negatively charged, so it's now negatively charged, and the sodium lost a negative electron, so it becomes positively charged. Anything with charges or anything that's really large can't get through this plasma membrane on its own. It'll need help from transport proteins, and that's what these are right here. Transport proteins. Now there's a whole bunch of stuff embedded within the phospholipid bilayer, not just transport proteins, but other important structures as well, which we'll talk about. So here, the membrane can allow for things to be transported in and out of the cell, can act as an anchor, right? It can attach to other surfaces. There are receptors that are found on the plasma membrane, and there are even enzymes. So there's a lot of different components associated with the plasma membrane. It is very, very, very important. I've already mentioned selectively permeable, meaning it chooses what can go in and out, and this kind of supports what I was mentioning before. Small substances, non-charged, meaning non-polar, can move in and out on their own, like lipids, versus ions and large substances, they need a transport protein to get in and out of the cell. We refer to the plasma membrane as what we call a fluid mosaic model because the proteins that are embedded in the membrane actually move. So if you think about a cell for a second and say, all right, on the left-hand side of the cell, there's all these proteins that need to leave, that need to exit. So what's easier? Is it easier to move all of those proteins? Let's say there's 150 of them that need to leave the cell. Is it easier to move all 150 of them to the other side of the cell where there's a transport protein? Or simply move a transport protein to where all of those, that 150 proteins are sitting? It's easier to move a transport protein there. So this protein relocation allows for faster movement of things in and out of cells. So the proteins will relocate to the most important areas where they are needed the most, which is really neat. It's a very interesting thing to see. So let's talk about the cell wall now. To understand the cell wall, we have to understand its composition. There are two major types of cell wall structures, gram-positive and gram-negative. The way they're structured, it's easy to tell them apart once we stain them. There are some atypical cell walls that we'll talk about later on, like Mycobacterium tuberculosis, an organism that causes TB, has something called a mycolic acid cell wall, which is different than a gram-positive or a gram-negative. There's a little bit of overlap, but still relatively different. Gram-positive cell walls have their plasma membrane, and then around them they have a thick layer of what we call peptidoglycan. Okay, so it's a very thick layer of peptidoglycan, which I'll show you in a minute what makes the peptidoglycan, what it's made of. And then that's it. There's nothing else outside the peptidoglycan. Gram-negative cells, on the other hand, they have the plasma membrane, and they have a thin layer of peptidoglycan, and then they have a thicker layer of what we call LPS, lipopolysaccharides, meaning it's made up of mostly lipids and sugars. So it actually has 3 layers instead of 2, like a gram-positive cell. So here's the big difference. If we are looking at them, in this manner, you see our plasma membranes. Both of them have that. Makes sense. Then the next layer, if it's thin, and then you have an outer layer, we consider that gram-negative. If you only have the plasma membrane and then one outer layer that's a thick peptidoglycan, then right then and there that we can tell that that's gram-positive. Staining these bacteria is actually clinically useful. Knowing whether an organism is gram-positive or gram-negative is extremely useful in a clinical setting. It actually will potentially alter the way you treat an infection. If an organism is gram-positive versus gram-negative, you may treat them differently because these structures act in a different manner. So depending upon the cell wall structure, whether it's gram-positive or gram-negative, we may treat an infection differently depending upon that structure. So let's look at these cell walls of gram-positive and gram-negative. So the peptidoglycan is mostly made up of two repeating sugars. They're called NAM, NAM, and NAG, NAG. So NAM is N-acetylmumeric acid, and NAG is N-acetylglucosamine. But I don't expect you to know the full names. Just know that it's made up of these repeating sugars, which are then linked together by these not only teichoic acids, but also these cross links right here. Okay, so you have these side chains. So you have teichoic acids and then amino acid side chains linking them all together. It's kind of like Lincoln Logs. You guys might be a little young for this, but instead of Legos, there were these things called Lincoln Logs. And they were simply pieces of wood that had grooves cut out in them. And pretty much the only thing you could make was a cabin. And that's what these things do. They cross each other over and over in repeating patterns and allow for this thick peptidoglycan layer to form. Now, if we were to compare that to a gram-negative cell, we would see how we have the inner plasma membrane, the thin peptidoglycan, and then the outer LPS, what we call the lipopolysaccharide layer. Okay, which is mostly, I mean, it's in the name, lipopolysaccharide. Lipo is lipids, and poly means many, saccharide means sugar. So it's made up, this outermost layer is made up of lipids and sugars. Now, again, knowing whether something is gram-positive or gram-negative may determine how we treat these infections. You may treat a gram-positive organism differently than you do a gram-negative in a clinical setting. So pros and cons of these two types of cell walls. Well, having a thick peptidoglycan layer gives a lot of protection. Having an LPS layer allows for the cell to have another level of A, protection, and B, selective permeability. So there are positives and negatives to both of these. LPS is useful but can be wiped away depending upon certain factors. The peptidoglycan layer are gram-positive, depending upon the organism you're looking at. Let's say you're looking at an infection with E. coli. And for that, most of the time, a person would be given penicillin. Why? Because penicillin inhibits the cross-links in the peptidoglycan layer. So you can see it specifically attacks different structural components. This is how a gram stain is done. In lab, we would normally do this within the first few weeks, but you will have a lab simulation, an online simulation of gram staining. First, you add the sample to your slide. You heat fix it, meaning you basically melt it to the slide. You add a purple dye called crystal violet, which gets into the spaces between the layers of the all those layers of the peptidoglycan, and it gets trapped. Okay, and then it also enters the LPS of a gram-negative. You add iodine after that. Iodine will attach to the crystal violet, making it bigger. So let's say this is the size of the crystal violet that goes into the peptidoglycan. So we know that the hole that this entered into is large enough for this to exit. But if we add iodine and it comes and attaches, now this is too big to exit the same way that it entered, right? So iodine acts as what we call a mordant, basically forcing the crystal violet to be stuck. It attaches to the crystal violet, and now the crystal violet is stuck. Then we use, it says acetone here, acetone alcohol solution. We don't necessarily use acetone all the time. We use ethanol, 70% ethanol. It's just another different type of alcohol. We use that to wipe away any excess dye on a gram-positive. And if you have a gram-negative cell, what's going to happen is the alcohol solution will literally wipe away the lipopolysaccharide layer, the LPS. It strips gram-negative cells of the LPS. And then we add one more stain after that called safranin. Safranin's a reddish, pinkish stain. And if you're gram-positive, you're already stained purple, and the spaces where the dye could go are already filled. But if you're gram-negative, what have we now exposed since the LPS is wiped away because of the alcohol? We've exposed the very thin peptidoglycan layer in a gram-negative cell. So then the safranin gets stuck within that very thin peptidoglycan layer in a gram-negative. So now when we go to visualize them on a microscope, you see either purple or pink, gram-positive or gram-negative. And depending upon the structure of the cell wall, will determine what color they actually stain. So if they're gram-positive, they'll stain purple. If they're gram-negative, they'll stain pinkish red. You will see that in your simulation. Errors can happen. This is extremely common. If you were in class, if we were all face-to-face doing gram staining, I promise you, many of you would mess up. Because sometimes I mess up. If you put too much of a sample on the slide, the stain may not stick. If you decolorize, meaning if you leave the alcohol on there for too long, it will wipe off everything instead of just the LPS layer. So your gram-positive will actually look gram-negative. So we can mess these things up pretty easily, to be honest. And it's very common. It just takes practice to get a repeating, getting repeated correct data from gram staining. Now, gram staining can tell us arrangement, morphology, but it really doesn't identify the bacteria. We can guess on a few of them. For instance, Staphylococcus aureus. Sure, we can guess that, or we can assume it's Staphylococcus because they're in clusters and they're round, but we don't really truly know what they are based on just gram staining alone. Gram staining is great for determining gram-positive versus gram-negative, so outer cell wall structure, also morphology and arrangement, but it's not helpful for identification purposes. So here is just talking about gram-negative versus gram-positive again. I'm not going to really go into this slide because I've already talked about it. So gram-negative species are typically harder to kill with a chemical agent than gram-positive because of that LPS layer. It is really, really good at blocking things from entering the cell. So it's this extra level of protection. The outer membrane, the LPS, can actually prevent damage from what we call lysozymes, which are enzymes, many different types of antibiotics. And some of the detergents and disinfectants we use are not effective at specific concentrations. So for instance, when I say we use 70% ethanol for gram staining, that's fine. But if we were to decrease it or or increase the concentration, we could have different effects. That's why when you use a cleaning product, not all of them are the same. Concentration matters. So some detergents and disinfectants in lower concentrations are not effective against gram-negative organisms. Porins, basically these are channels in the outer membrane, in the LPS, that allow things to enter the cell. So porin is simply a protein channel that allows things in and out of the cell. And they're nonspecific. Con of a gram-positive cell wall, it is more sensitive to compounds that interfere with peptidoglycan. I've already mentioned penicillin. Penicillin can easily destroy any cell wall that's being built. It actually prevents the linking of the peptidoglycan. Some pros of gram-positive: the fact that it's thicker, it can help maintain moisture longer, provides protection from mechanical stresses. That's more of like environmental stress, heat, pH, things like that. And then the presence of those teichoic acids that I've mentioned help stabilize the wall. So even though they're overlapping like this, everything can shift. It can move. It's not a static, rigid structure. It can kind of shift as necessary. And it'll also help maintain the shape, the overall shape of the organism. So this is a table 3.1, comparing gram-positive to gram-negative. The important things for right now are peptidoglycan, the staining color, and then also penicillin susceptibility. I've already explained that penicillin, gram-positive are more susceptible because penicillin attacks the linkages between the NAM and NAG peptidoglycan layers. These other things, lipids, porins, outer membranes, we'll focus on those later on, especially the lipids and the fact that it's an endotoxin. We'll focus on that in a different chapter. Transport. So cells have to move things in and out of themselves, right, on a constant basis. They need things to move in, and then they need to get rid of waste, for instance. So there are two major types of transport: passive and active transport. Passive transport requires no energy investment, meaning things move passively and no energy is required. Active transport, on the other hand, requires energy. Okay, it requires energy. Let's see some examples. First example is diffusion. So when we talk about passive transport, there are several things we need to think of immediately. One, passive transport means things or molecules are moving from high to low concentration. Passive transport means a substance is moving from an area of high concentration where there's lots of it to an area of low concentration where there's very little of it. Okay, and in this image you can see the green, there's lots on the outside, and then there's very little on the inside, and they're just moving through from high to low. That is going to be the case in every type of passive transport, from high to low concentration. Now, the other things we need to take into account are the chemical nature of the substances. Since these green ones can move directly through the phospholipid bilayer, then they must be either nonpolar or really small, or both. Okay, so nonpolar substances, I've already mentioned this, can pass through the membrane without help. So these substances passing right through, they're nonpolar. and or they're really, really, really small. On the other hand, by the way, that's called simple diffusion. That's the most basic form. It's called simple diffusion. If you get a little bit more complex when you go to facilitated diffusion, still going from high to low concentration, however, there is a transport protein that's involved. A transport protein spans the entire plasma membrane, and it lets things in and out of the cell. You can see here going from high down through the transport protein inside to the low. This is facilitated diffusion. Now, why would they need a transport protein? Right? Well, what would make sense to the point where they would need a transport protein? What does that imply that they are? Well, it implies that there are a couple things. It implies that they are polar, meaning having a charge, or ionic, and or really large. Those three substances or conditions require facilitated diffusion, require help of a transport protein. If something is large, if it's ionic, meaning it has a charge, a full charge, or polar, meaning it has a partial charge. So charges, whether it's charged or whether it's really large, if it's moving down the gradient through a transport protein, that is facilitated diffusion. Okay. Still, no energy. It's all passive. No energy required. So this is just two definitions: simple diffusion, small, non-charged molecules, right? Non-polar, lipid-based for the most part, and then facilitated diffusion, basically anything that has a charge to it requires, or is really large, requires a transport protein. Another situation, and this is a special case of passive transport, it's called osmosis. Osmosis is the movement of water. Now, it's the flow of water from area of high concentration to low concentration. So it's the diffusion of water. Now, when studying for this type of thing, when I was in undergrad and actually in high school, I learned a little bit of a different way to think about this. So when people say it's osmosis, the diffusion of water, I always remember it as water follows solute concentration. What do I mean by that? I mean, if you have a membrane, and on one side you have lots of salt and very little salt on the other side, water is going to move from the area of low salt concentration to the area of high salt concentration. So it follows solute concentration. The examples on the screen are really great examples. So I'm going to start from right to left. Isotonic environments mean the inside of the cell. Let's say we're referring to salt. The inside of the cell has roughly the same concentration of salt that the outside of the cell has. Let's say there's 5% inside and 5% outside. Because of the fact that the concentrations are roughly the same, the amount of water that leaves and enters is about the same as well. Our body, for instance, is an isotonic environment. Basically, our cells, there's just this movement of water in and out of them because it's balanced. If it's unbalanced, then we have some problems, which are the next two. So in isotonic things, there's really no water loss or gain. It's just kind of moving in and out. Hypotonic, the way I always remember that is that when you say hypo, things get bigger, and then it's going to eventually burst. So what this tells us is there is more of a salt, we're still using salt as an example. This could be anything. It doesn't have to be salt, but we're going to use salt as an example. There's more salt inside the cell than there is in the water. So if water follows solute concentration, tons of water will rush into the cell and it will eventually burst. It'll eventually burst. So there's a higher concentration of solute inside the cell than there is outside in a hypotonic environment. The cell will then rupture. In a hypertonic environment, it's the opposite. There's a higher concentration of solute outside the cell than there is inside. So in a hypertonic, the cell actually collapses. The cell actually collapses. It undergoes what we call plasmolysis. Okay, it undergoes plasmolysis. The cell will collapse and it will just fold in on itself and it'll just die. This is honestly really interesting because in a hypertonic environment, before there were freezers, before we had freezers and electricity, we would store food in ice chests, and what we would do is we would throw salt or sugar on it. Now, people are always, why would you throw salt on the food? Well, the salt, when you salt food, it creates a hypertonic environment. So when you're trying to store food for extended periods of time, they threw salt on it to create a hypertonic environment, and any bacteria on the meat would undergo plasmolysis because it just created a hypertonic environment by simply adding salt, just throwing handfuls of salt on top of the meat, and it would allow it to preserve the meat for a longer period of time because the bacteria that were living on the meat and that would have eventually destroyed it and kind of broken it down would have died because of the hypertonic environment. Very interesting that something so simple actually is a huge mechanism to make sure food stays longer. Granted, now we have fridges and freezers and all these cool gadgets to prevent that from happening, but back before electricity, they had ice chests, they would throw salt on it and sugar on it, and it would create a hypertonic environment, and the bacteria living on the meat would die. They'd undergo plasmolysis. So, basic concept, really cool to see in action. So, hypertonic plasmolysis, cell collapses and dies. Hypotonic, it ruptures. Isotonic, it's an equal balance and it just moves in and out. There's no net gain or loss. These are the three types of passive transport, none of which require energy. Now if we move to active transport. Active transport requires a couple things. First off, it requires energy, specifically ATP. Second, it requires a transport protein. And third, the third characteristic is the fact that it moves against the concentration gradient from low concentration to high concentration. It is going in the opposite direction of passive transport. So it's going from low to high instead of high to low. All of these things are characteristics of active transport. Requires energy, requires a transport protein, and it moves, whatever you're moving is going against the gradient. Whatever you're moving is going against the gradient. Think of it this way. You're in an inner tube or a raft going down a river. You're not paddling, you're not doing anything, you're just kind of sitting there, relaxing, and letting the current take you, right? You're not exerting energy, you're just going with the flow, right? Same thing with passive transport. They're just going down the gradient. There's nothing really to do. There's no energy expenditure. But let's say you're going down the river and all of a sudden you realize you missed your exit, right? The landing you were supposed to get off of. And then all of a sudden you just jump into the water and start swimming upstream to get back to that landing. So now you're going against the current or against the gradient, right? If we're comparing. So you're going against the current. You're expending a ton of energy in the process as well. So active transport requires energy, requires a transport protein, and you're moving against the gradient. This is a really good mechanism here. There are three main classes of active transport. We are only going to talk about primary. All we are talking about is primary, and we're talking about it in the most simple form. Goes against the gradient, uses a transport protein, and it costs energy. It takes ATP. Okay, I do not expect you to go into secondary and tertiary active transport. We're only talking about primary. Okay, so in your book, ignore it. We've mentioned some external structures for bacteria before. We mentioned glycocalyx, we mentioned flagella, we mentioned capsules. So those are some extra structures that they have on the outside of the cell in order to allow them to do certain things. If we look inside, the nucleoid region is basically this whole red region right here, and it's where the chromosome is. So wherever the chromosome takes up space, that's what we call the nucleoid region. So previous slide, we talked about external structures, movement, adhesion, protection. Perfect example of movement, flagella. Adhesion would be slime layer, protection would be capsule. Okay? Here on the inside, now we're talking about the nucleoid, where you find the single circular chromosome that the bacteria has. You can see it's in red here. This is the nucleoid region. Some bacteria can form endospores. This is actually really important. Endospores are essentially metabolically inactive cells. So, for instance, the two major genera that form endospores are Clostridium and Bacillus. Okay, Clostridium and Bacillus. Now, I know I also said Bacillus was a shape. Yes, the name has two meanings. You could be talking about a specific genus, Bacillus genus, or you could be talking about the shape. So for the most part, I'll probably refer to cells as rod-shaped bacteria instead of bacilli-shaped. But the two genera that form endospores are bacilli and Clostridium. What happens is they're rod-shaped, and what they will do is they will move all the genetic material to one spot in the cell, and then they'll form layer upon layer upon layer of protection around it. And they do this when the environment is not favorable. There's little to no nutrition. They're being outcompeted for space or resources, among many other things. So they'll move all the genetic material to one area, they'll surround it over and over and over and over with protective layers, and then the rest of the cell dies, and you're just left with a little spore. If you've done chapter 1 or lab 1, and you did the simulation, you had to look at endospores in the microscope in one of the simulations. Those endospores are one of the most difficult structures to destroy in all of microbiology. Endospores are hard to kill. They're the ones we really have to worry about when it comes to, for instance, getting a tetanus shot. So Clostridium tetani, for instance, it forms endospores. Let's say on a rusty nail, it runs out of nutrients, it runs out of space, so that it's just hanging out there in its endospore form. It's just a giant circle with just genetic material in it. Then you step on that nail, and it penetrates your foot. Now the endospore is in a very favorable environment, so the cell will actually germinate. The endospore will open back up, and you'll germinate into a whole new cell. So they can actually go back to endospores, and then when it's favorable, germinate and be an active cell again. This is extremely, extremely difficult to destroy. To destroy endospores, you have to use very powerful mechanisms like a machine called an autoclave, which we'll talk about later on, or UV light, specifically gamma rays. So there are ways to destroy it, but it's very difficult, and they can survive pretty much anything. We have found bacteria living in ice core samples in the Arctic, because they formed endospores and just were hanging out as endospores until they were in favorable conditions and could germinate. So, yeah, the endospores are really difficult to treat, and we really want to make sure that we kill them when we're trying to, let's say, package food, for instance. If you ever buy a can of food and you set it down on a table and it rocks back and forth, like there's a little bow in the top or the bottom of the can, don't eat it. Because at some point, there were endospores in there, and then they germinated, and now they're in there replicating, and they're creating gas as a byproduct. So what happens is the can expands, and the top or the bottom will kind of bubble, and they won't sit flat on the table. So you may have, if you've ever had like Snapple or some types of Starbucks cold drinks that you can get from like the store, gas station, or wherever, they have those little clicky caps. The lids, they have the little clicking mechanism on them. And as a kid, you probably used to play with them and piss your parents off with them. Just me? Maybe. But you would sit there, and I'd sit there and play with them, click, 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 click. That actually serves a function. Its purpose is if gas builds up, it pops out. And you shouldn't consume that product, because when they seal it, there's nothing in there. If there's nothing in there, no gas will be produced, and it will stay in its lowered position, and it won't bow out and be able to be clicked. But if the lid is on and you can press it and it clicks, that means there's something in there producing gas as a byproduct, and there's gas pressing on that lid. You do not want to consume that, because you will get sick. Chances are you're going to consume some type of pathogen and get sick. So endospores, really dangerous. Sporulation, this is just how they form endospores. I'm not going to really ask you to memorize the steps. Basically, it's really similar to binary fission, except you're not making two new cells. You're actually just forming. You're going to copy the DNA, and then you're just going to package it all into this one big bubble, and then the rest of the cell dies. So I'm not going to ask you the steps really associated with this, but these endospores can survive so many different environments. They are very, very dangerous if they're pathogens, like Clostridium tetani, which causes tetanus, Clostridium botulinum, which causes botulism, which is food botulism, wound botulism, infant botulism, meaning there are different forms. Also, Clostridium perfringens also forms endospores, and that causes gas gangrene. If you don't know what gas gangrene is, look it up. We'll actually talk about it later in the semester, but basically, if it's Clostridium perfringens and it gets into your body, let's say a puncture wound in your thigh. It will germinate and it will destroy the underlying tissue. Your thigh will swell because there's gas building up underneath as it destroys your tissues. And then they cut it open and it's just dead tissue on the inside. It's all black, everything is dead. And in some cases, there's just no way to save that limb. If it's gone on long enough or progressed far enough, that limb is gone. It needs to be removed. If they save it, what they have to do is something called debridement, essentially scraping all the dead tissue out. It is honestly one of the craziest things I've ever seen. And we talk about it later in the semester. This is a perfect example of how it shows endospores functioning, how they form, and how they are then released. It all depends upon whether or not the conditions are favorable. The endosymbiont theory. So the endosymbiont theory is basically referring to the fact that in eukaryotic cells like human cells, the mitochondria used to be unicellular organisms. So millions of years ago, after several events, what we call sequential events, mitochondria, or a unicellular organism that was really good at making ATP and energy, was incorporated into a eukaryotic cell. So you got to think about it this way. Eukaryotic cells would destroy something and get a little bit of energy. Right? Consumed it, got energy. When we're talking about cells, we're not talking about organisms, we're not talking about multicellular organisms, we're talking about single-celled eukaryotic cells. So they would engulf it, and they would destroy it, and they get some nutrients. But after a bunch of cell merging events, eukaryotic cells, instead of destroying this unicellular organism that they brought into their cell, engulfed it, instead of destroying it, they said, whoa, wait a second, this thing is really good at making energy. It's way better than just eating it and destroying it and getting a little bit of energy. What if I keep it and let it make energy and I just use it? So after a bunch of these sequential cell merging events, eventually mitochondria were incorporated into eukaryotic cells. And there are quite a few ways that we can kind of prove this, or at least support it. There's also not only just the mitochondria, but for plants, a similar thing happened with chloroplasts, which are the organelles that allow plants to undergo photosynthesis. So here, mitochondria engulfed non-photosynthetic prokaryote. Chloroplast evolved from the photosynthetic prokaryote, cyanobacteria. This is exactly what I just said, just written out. Now, here is just kind of a really interesting way to look at it. Here is your non-photosynthetic prokaryote, the one that's really good at making energy. Here's the organism, the before it was a eukaryote, it was a proto-eukaryote. I'm not going to get that specific, but very primitive right here, non-photosynthetic eukaryote. And it incorporated this cell, and now going forward, it used that cell to produce as much energy as possible. And then over here, you can see the divergence of where plant cells incorporated. Now, these are primitive photosynthetic cells, very primitive, because we're talking billions of years ago. But they incorporated photosynthetic bacterium and then just said, hey, they're great. They make lots of energy for us. Let's keep them. And they were eventually incorporated into the cells. Now, how do we know this? The question becomes is how are we so sure, or pretty sure, that our mitochondria were at one point in evolutionary history prokaryotic cells. And there's quite a few reasons. First off, they have their own circular DNA. So if you looked at our mitochondria in our cells, they have their own circular DNA, like bacteria. They have ribosomes, what they call 70S ribosomes. That just refers to the structure, the 70S. But the ribosomes inside the mitochondria are closer in similarity to those that are in bacteria than they are to human ribosomes. Human ribosomes are quite a bit different than the ones that are associated with the mitochondria and bacteria. It also has a double membrane. The mitochondria has an outer membrane and then a highly folded inner membrane. Also has a double membrane. It's similar in size to bacteria as well. It's relatively small, which makes sense that proto-eukaryotes or primitive eukaryotes could engulf them and keep them in the cell because they're so much smaller. They have the ability to replicate similar to binary fission. And then the genes resemble certain bacterial genes. So there is a ton of evidence to support the endosymbiotic theory that mitochondria and chloroplasts at one point in evolutionary history were their own individual prokaryotic cells that eventually, after some sequential cell events, they were incorporated into primitive eukaryotic cells. For some, it was the mitochondria, and only the mitochondria, because they were non-photosynthetic. But photosynthetic cells, primitive eukaryotic photosynthetic cells, also got not just the mitochondria, but they also got the chloroplasts as well, because it allows them to carry out photosynthesis. Very interesting. Lots of evidence to support this. So that is the end of this chapter. Please take your time and watch it a couple times. Watch this video a few times. This way you cover all the information that I went over. There's a lot, even though this is only a 43 slide PowerPoint and only an hour-long video. There's still a lot of information. So what I recommend is look at the PowerPoint, then also look at or watch the video, and then look at your book, because it's going to be important that you understand the basis for a lot of this material going forward. Morphology and arrangement, very important. Endospores, very important. Fluid mosaic model, the ability to move stuff in and out of cells, all that, super important. So if I focused on it in the PowerPoint and in my lecture video, read it in the book as well. Okay? I hope everyone has a nice day, and I will see you all soon. All right, have a good one. Stay safe.